Good morning, everyone. In this session, we will talk about immunomagnetic sorting of plasma cells. The importance of plasma cell sorting. Since the myeloma-associated cytogenetic abnormalities are limited to the plasma cells, morphology of the plasma cells are not discernible after the processing for FISH testing, and hence a prior sorting or plasma cell enrichment is necessary to ensure that we are looking only at the plasma cells and not on other cells. Thus, the sorting improves the sensitivity of detection of these cytogenetic abnormalities. This is especially important when we are looking at the marrow, which has very low percentage of plasma cells. In the conditions like very early in the disease, like in MGUS, and also in post-therapy samples. There are various techniques for the enrichment of the plasma cells. To enlist, we have magnetic cell sorting, we have fluorescence activated cell sorting, targeted manual scoring of plasma cells, customized automated image analysis, and kappa and lambda immunofluorescence staining, which is followed by FISH testing. Among these techniques, the magnetic cell sorting is considered the technique which has maximum yield and is also the cost effective. Coming to the magnetic cell sorting, it is broadly of two types. The first is the column-based technique, and the second is the column-free technique. The column-based technique is marketed by Miltenyi Biotech, and it includes three important steps. The first step is to label the cells with the antibodies and magnetic beads. After this, the cells are passed through a column which is kept inside the magnet. The labeled cells remain inside the column. The column is then taken out of the magnet, and subsequently it is followed by elution of these labeled cells. Finally, these eluted cells can be used for downstream applications like for FISH testing and for molecular genetic analysis. The second technique is the column-free technique, which we will show in the subsequent workshop. In this column-free technique, again we add the antibody and the magnetic bead particles. This is followed by the putting of the tube inside the magnet and then a period of incubation. After the incubation, we can pour off the cells and collect the cells according to two important techniques. One is the negative selection, in which the desired cells are decanted into a fresh tube, and we have the positive selection, in which the desired cells remain inside the tube and they are sticking at the sides of the tube. The plasma cell sorting is affected by various pre-analytical variables. In our experience, we have found discrepancy in plasma cell percentage based on the first pull and subsequent bone marrow aspirate samples. So it is recommended that during the procedure, after making of the bone marrow aspirate slides, the next sample should be given for cytogenetic analysis in a green top vial. And subsequently, we can use the next pull samples for other techniques like for flow analysis. So the sample should be collected in a green top vial which contains sodium heparin as an anticoagulant. It should be 2 to 3 ml minimally diluted sample. And as I already said, it should be preferably from the first pull or after relocation of the needle. The tube should be inverted 5 to 6 times to avoid rapid clotting of the aspirate sample and adequate mixing of the anticoagulant. The sample should be transported at room temperature rapidly to the lab. There are certain conditions in which plasma cell sorting is of limited value. These include when we have a dry tap sample. In such conditions, imprint smears are preferred for subsequent analysis. Also, since the plasma cell myeloma is a patchy disease, we can have very less number of plasma cells on one side of the aspirate, but the other side biopsy can show large aggregates of plasma cells. So in these conditions, we have to use the imprint smear which is made from the biopsy sample. We can mark the areas which contains large aggregates of plasma cells, followed by destaining of these imprint slides, and then these imprint slides can be used for fish testing. Now we will proceed to a practical demonstration of plasma cell sorting. Thank you.